It all starts at the primitive mouth of a 5-week-old embryo. At the location of the future dental arches, the epithelium of the primitive mouth is called odontogenic epithelium. Under this odontogenic epithelium, the mesenchyme is turned into ectomesenchyme, arising from neural crest cells. The odontogenic epithelium then sends induction messages that convert the neural crest cells into odontogenic cells. Then, by shift of the mitotic spindles of the basal cells, the odontogenic epithelium proliferates and then becomes invaginated, forming a U-shaped band known as the primitive epithelial band. This primitive band bifurcates into a primary dental lamina on the inside and a vestibular lamina, which will eventually form the vestibular mucosa on the outside. It is on this dental lamina that the teeth will form. For this to happen, dental placodes will appear, inducing the formation of dental buds. The ectomesenchyme then sends signals that trigger localized cell proliferation. The bud thus turns into a dental cap. This cap is formed around the dental papilla. It is surrounded and protected by the dental follicule. The germ of each deciduous tooth is thus formed with a certain time lag. After this, we reach the bell stage. The tooth germ increases in volume and expands while creating the future enamel-dentin junction. Finally, the coronal dentin and enamel of the tooth are progressively formed along the cusp slopes. At the end of the crown stage, the enamel is completely formed. It is covered by the reduced enamel epithelium that protects the enamel of the dental follicule. All this lodged within the bone crypt. The root's formation starts with the proliferation of the epithelial cells of the cervical loop, located in the region of the cervical margin of the crown. This creates an epithelial sheath, which bends inwards to form Hertwig's epithelial root sheath. Hertwig's epithelial root sheath is a bilayered cellular structure composed of an inner epithelial layer facing the dental papilla and an outer epithelial layer facing the dental follicule. The apical bent portion of the sheath is named epithelial diaphragm. Its role is to induce the radicular dentinogenesis. For this to happen, the diaphragm sends induction messages to the ectomesenchymal cells of the apical papilla. These messages induce the differentiation of the ectomesenchymal cells into odontoblasts. The newly formed root odontoblasts then secrete the radicular mantle dentin in contact with the inner cells of Hertwig's epithelial root sheath. Along the dentin matrix thus formed, the cells of the outer epithelial layer disintegrate, leaving only the inner epithelial layer. The cells of this sheath then synthesize and secrete enamel-related proteins before detaching from the root surface. The mantle dentin and enamel-related proteins come thus directly in contact with the dental follicule, inducing the primary cementogenesis. For this purpose, cells of the dental follicule react to these induction signals and differentiate either into fibroblastic cells that migrate towards the root surface with a perpendicular orientation, or into cementoblasts placed parallel to the surface of the tooth. Then, the fibroblastic cells secrete the extrinsic fibers, and the cementoblasts secrete the intrinsic fibers of the primary cementum. And that is how the dentin and the primary cementum of the root are formed.